Welcome everyone. My name is Alejandro Berenguer Rubio and I am a predoctoral researcher in the Cytogenetics lab at La Fe Health Research Institute in Valencia, Spain. And it's a pleasure for me to share the work that I'm doing and where I'm developing my PhD. So today I will talk about optical genome mapping as a powerful cytogenetic tool in soft tissue and bone sarcoma. So let's start. Okay, and I think that for the introduction, it's interesting to briefly introduce my group. So in the La Fe Cytogenetics laboratory, we have been working with Bionano and with optical genome mapping for 4 or 5 years, mainly with hematological malignancies. But since mid-2024, we started a new project based on solid tumors, especially sarcomas, in collaboration with Dr. Parmayordomo, who is a specialized pathologist in this field. We have analyzed more than 100 samples of sarcomas, and I'm going to share the results with you. So, I think that it's time to answer that big question of what are soft tissue and bone sarcomas. And, well, you can Google it yourself, but at the end, sarcomas are rare malignant neoplasms that arise in the soft tissue and in bone. They are considered to be rare cancers because they represent less than 1% of all adult malignancies. And I have to say that in addition, they are heterogeneous as well, because we know more than 100 subtypes of sarcomas. So this rarity and the heterogeneity pose a major challenge in diagnosis, research, and the development of effective targeted therapies. So now we are going to speak about the current diagnosis of soft tissue and bone sarcomas. And in the workflow, everything starts with the biomedical imaging, and this will guide the tumor biopsy. And with this biopsy, the pathologist will evaluate both morphology and immunohistochemistry of the sample, and then specific alterations can be found using FISH and NGS techniques. And then finally, definitive diagnosis is made following the WHO 2020 classification guide. So then we can talk about the diagnosis of soft tissue sarcomas from a genetic point of view. So despite all this workflow, the sarcoma diagnosis is still challenging. And speaking about genetic, we have two mainly groups of sarcomas. On the one hand, we have sarcomas with simple alterations and defining genetic alterations that have a high diagnostic value with consistent morphology, and they are, for instance, Ewing sarcoma, synovial sarcoma, and they represent around 20% of all cases of sarcomas. Then we have, on the other hand, the sarcomas with complex karyotypes, with no defining alterations, with very heterogeneity in morphology, and they're often considered undifferentiated sarcomas. But they can be liposarcomas, myxofibrosarcomas, and osteosarcomas. Okay, so in total, they represent around 80% of all cases. And another thing that I have to say, and it's important to note, is that the sarcomas with simple alterations can evolve to sarcomas with more complex karyotypes. They can suffer a process of differentiation and they become more aggressive. But the underlying mechanisms are still poorly understood. Okay, so as a result, many questions remain unanswered and therapeutic options are still limited. So now, we go to the hypothesis of our work, and given the current limitations in sarcoma diagnosis, prognosis, our current hypothesis is that the integration of optical genome mapping with other techniques such as epigenetic approaches, sequencing approaches, or transcriptomic techniques could in the future enhance the diagnosis of sarcomas by improving the identification of relevant findings in these tumors that will perhaps in the future contribute to enhance our understanding of the molecular mechanisms underlying this disease. So right now, in order to achieve that goal, we need to follow several objectives. And the first one is analyzing with optical genome mapping 100 samples of sarcomas retrospectively, and then 300 samples prospectively. Okay. Then we will compare OGM results with those obtained with standard of care techniques to see if they match with the results. Okay. Then another step is we will integrate the OGM data with other data, like as I said before, epigenetics, transcriptomics, sequencing, and at the end we will correlate this OGM and genomic data with clinical data to see the outcome of the different patients and to see if that the information match. Okay. So now I will talk about materials and methods, but to be honest, I will not go into much detail here because I assume that most of you are familiar with optical genome mapping and Bionano technology. And in brief, OGM represents a next generation cytogenetic tool that allows the unbiased detection of structural variants and copy number variations all across the genome, okay, the entire genome. So this is achieved by extracting ultra-high molecular weight DNA molecules that will be labeled, then will be loaded onto Bionano chips, and the molecules will be linearized into nanochannels. And then the safer system will take pictures of that molecules, and at the end we can see the results. Okay. So in this work, we have the following workflow, and the samples were obtained from biopsies and tumor resections. They were initially snap frozen in liquid nitrogen and stored at -80 degrees Celsius, enabling then the extraction of ultra-high molecular weight DNA. And then the results were analyzed using the rate variant pipeline. Okay. So now I'm going to talk about the results, but as this study is still ongoing and the results have not yet been published, I will mainly focus on representative examples of different sarcoma subtypes and cases where OGM contributed to the understanding of the molecular mechanisms behind sarcomas. Okay, so let's start with the first group of sarcomas that are high-grade myxofibrosarcomas. I know that the name is long, but myxofibrosarcomas are tumors that originate from connective tissues. Okay, and we can see here that they have a very complex karyotype, but there are no defining alterations. So we have several different mutations, several different alterations, and we can see here multiple CMVs, multiple structural variants. But it's interesting because we observe a trend to subgrouping, okay, the myxofibrosarcomas based on the number of alterations. So we can see that less aggressive myxofibrosarcomas have a less number of alterations than high-grade that are very aggressive myxofibrosarcomas, and they have high number of alterations with aneuploidies, rearrangement, as you can see here in this slide. And at the end, the main question here, or main goals, are to see the alterations that are related to a bad outcome, and to see if we can create categories based on OGM profiles. Okay, so we are starting to see that subgrouping. And now we are going to talk about undifferentiated pleomorphic sarcomas. And this group of sarcomas is a very heterogeneous group, because I have to say that they are often considered a diagnosis category of exclusion. So this is when a pathologist looks the sarcoma, and they observe that there is a high-grade sarcoma, very aggressive, but they don't match this sarcoma to any of the diagnostics. an UPS diagnostic is made. Okay, so we have here a very heterogeneity group with complex karyotypes as well, with multiple copy number variations, multiple structural variants, but non-specific alterations. Okay, so mainly we here the questions and the goals are the same to match these alterations that are related with a bad outcome and to do categories. But in this case, we have we think that there are categories that exist because at the end, as I said before, they are very heterogeneous group. Okay, and we have sarcomas that have different origins. So we think that looking at OGM alterations, we will find subgroups inside of this group of undifferentiated pleomorphic sarcoma. Okay, right now I'm going to talk about liposarcomas, and liposarcomas are sarcomas that arise from the adipose tissue, so from fat. Okay, and there are multiple subtypes of liposarcomas, but we're going to focus on these three liposarcomas that are the well-differentiated liposarcoma, the typical lipomatous tumor, and the differentiated liposarcoma. And okay, if we follow the WHO classification, well-differentiated liposarcomas and the typical tumors are the same kind of sarcoma, but the only difference is that the well-differentiated liposarcomas localized in trunk and retroperitoneal space, whereas a typical lipomatous tumor are localized in extremities. Both affect to adipocytes, and the grade of aggressiveness is low to intermediate. Okay, but then we have the differentiated liposarcoma that can be localized everywhere in the trunk, retroperitoneal space, and extremities. And the different thing here is that adipocytes are affected, but these adipocytes don't remain as adipocytes. They suffer a process of dedifferentiation, and that makes the differentiated liposarcomas high aggressive sarcoma. Okay, so we can see here that they have complex karyotypes, as you will see later. There are multiple CMVs, multiple structural variants, but there is a specific alteration that is MDM2 amplification together with CDK4 amplification. And there are some studies that shows that MDM2 amplification is correlated with prognosis of patients. So we can see that when the grade of MDM2 amplification is higher, the prognosis and the outcome of the patient is worse. You can see here in this graph. And here we have the karyotypes of liposarcomas. We can see obviously that the differentiated liposarcomas have more complex karyotypes than well-differentiated liposarcomas. And our main goals, our main questions here are the same as before. And now I'm going to talk about the fourth type of sarcoma, that is the high-grade osteosarcoma. And as the name said, osteosarcomas are the most common type of bone sarcomas. There are different subtypes of osteosarcomas, but mainly they harbor non-specific alterations, okay, and very complex karyotypes with multiple copy number variations, multiple structural variants, and there are no specific alterations mainly. And I say mainly because there are a lot of kinds of osteosarcomas. We have the conventional ones that are high grade with non-specific alterations, but then we have another type that is low grade osteosarcoma, and is called parosteal osteosarcoma, that harbor the MDM2 amplification as liposarcomas. But anyway, here I just want to show you these complex karyotypes of osteosarcomas and our main questions here that are the same as before. We want to see if there are alterations that match with a bad outcome, and if we can make categories based on OGM alterations. So right now we are going to speak about Ewing sarcoma. This is the last type of tumor that we are studying, and Ewing sarcoma can affect both bone and soft tissue. and they are often characterized by that aspect of round, small cells, okay? And talking about genetics, there is a specific alteration that is this gene fusion. Is called EWSR1 with FLY1, okay? And this is because of the translocation of chromosomes 1 and chromosome 22. And thanks to OGM, we saw different alterations that are coexisting with this known gene fusion. And right now, our main question here is if these extra alterations that are together with this gene fusion have an impact with outcome. Okay, so we can associate that extra alterations with a bad or a good outcome, and to make categories based on OGM alterations as well. So right now, I want to tell you, and this is an example, okay, of how OGM is useful and can be the ideal method to see the underlying mechanisms of the disease. This case is a case of a 25-year-old with Ewing sarcoma. We can see here that the standard of care techniques, a small round cell sarcoma morphology, but the FIS failed to identify the Ewing sarcoma fusion, okay? So we did an OGM, okay, and the result is this. We can see here a very complex karyotype with multiple aneuploidies, with multiple translocations, but the most important alteration here is this gene fusion that is MN1 with CXXC5. And this is a fusion that has already been described in astroblastomas and recently has been described by some colleagues, and now we have this fusion described in sarcomas. So this is an example of how OGM can improve the understanding of the molecular mechanism behind sarcoma. Okay, so now these are the results, and now I want to tell you a little bit about the future directions of this project. So as I said before, we want to cover all biological layers. So in the future, we will do a whole genome sequencing or maybe whole exome sequencing. We will perform transcriptomic studies and epigenetic studies. And once this part has already been done, when we complete all the omic data, will pass to the molecular studies. Okay, so we will use mainly cell lines, we will use animal models, organoids, to see if this signature of genes that are affected, we can correlate that in that animal models, and we can test some experiments. So these are our future directions in this project. So now I want to tell you about the study limitations of this study. And well, the first one is pretty obvious, and I think that it's important because at the end all pathology labs works with paraffin samples. So for extracting ultra high molecular weight DNA, we need frozen tissue. So I think that one important point is to freeze more amount of tissue. Okay. Then in the second limitation, it is related to the other one, and this is the sample size. When we do a biopsy, there's a great amount of tumor, and then there's a limited amount of tissue that is for research use. So we often have a limited sample size. Okay. Then the third study limitation is the storage, because at the end it requires a minus 80 degrees, and this is space consuming and very expensive as well. And at the end, sarcomas are rare diseases, so rare cancers. So our core size is small, so we need consortia in order to increase our core size. And this is a studio limitation that is very important as well. Then these are the main conclusions, the take-home messages that you need to sum up all the thoughts that we have been talking about. And just I want to thank all the agencies that are funding this project, and thank you all for your attention and your time.